So in this chapter, we're going to be taking a look at the third line of our immune system response. So we're looking at lymphocytes specifically. We're going to talk about B cells or B lymphocytes and T cells or T lymphocytes. And we're going to talk about how they can recognize very specific invaders that get into our body and how they try to get rid of them out of our body. So we've already gone over first line of defense, which prevents or tries to prevent things from getting in the body. Second line of defense, going over the non-specific ways to get rid of foreign invaders. And this third line of defense, it has specificity. It is looking and recognizing for very specific things that gets in our body, and then learning how to get rid of it and having that memory and how to take care of it if we'd ever get infected again. So kind of the, scoping out the chapter, we're going to talk about what are antigens, the fact that we can recognize all of these foreign invaders. They don't have to be microbes, they could be different plant materials, they can be dust, things like that. We're going to talk about the different types of lymphocytes, those B cells and T cells, kind of how we activate those B cells and T cells to fight a full immune response. Yes, the T cells, the B cells. We're also going to talk a little bit about vaccines, kind of the different kinds of vaccines and how we develop that immunological memory. So, kind of starting off the chapter, just a little bit about what the third line of defense is. It's sometimes also called adaptive immunity or acquired immunity, and it's anything that has to do with your B lymphocytes or T lymphocytes, your B cells and T cells. And again, it's the fact that these cells could recognize millions, if not billions, of different types of molecules that could get in the body that are foreign. And it can recognize them as foreign, and it can mount a very specific immune response against them. Now, an antigen is any type of molecule that's going to stimulate an immune system response. So anything that the body pertains as foreign, that's going to stimulate some type of immune response, either by the T cell or the B cell or both. So the features of our adaptive immune system is that it has that specificity. The fact that we can recognize specific invaders, we can produce very specific antibodies to recognize those specific invaders in the future, allows us to now have memory against invaders. So if we'd ever encounter them again, our immune system knows how to get rid of them very, very quickly. In theory, you wouldn't contract whatever disease that antigen might cause. Now, a little bit about our immune system response. There are lots of things going on in the adaptive immune system. So I'm going to be talking about B cells and I'm going to talk about T cells a lot, but we're going to talk about, just differentiating the different parts of our immune system. We're going to talk about how lymphocytes and antigens recognize each other, the cooperation between the different lymphocytes because they do work together. And then we're also going to talk about how they can produce various types of antibodies and other types of T cell responses. So lots of different things. In your book, this is figure 15.1 that talks about a couple different types of our lymphocytes, our T cells, and they're called T cells because they mature in the thymus. Our B cells are called B cells because they mature in the bone marrow. They both like to hang out in lymph nodes, and again, and I'm like, they still circulate around in the body. They are there to patrol, looking for foreign invaders. So anytime, here's just an example of one of our T cells, that's a T helper cell, and I'm like, it can recognize foreign invaders, these little red-green things, and I'm like, we have different other types of dendritic cells that can recognize foreign invaders, and I'm like, they're constantly recognizing foreign invaders. Once they recognize them, whether it's a B cell, whether it's a dendritic cell, whether it's a T cell, we can activate them. Because we're not going to fight a war when there's nothing there to fight against. So we're going to activate those specific cells. So if this B cell recognizes something, it's now going to become activated, and it's going to make more copies of itself, because you're going to win a better war if you have more of yourself, if you've got more, a stronger army. And so activated B cells will make more of themselves. They're also going to make some memory cells. Activated T cells, same thing. They're going to make more of themselves as well as other chemicals, but they're also going to make some memory cells. So we're constantly trying to recognize foreign invaders, whether B cells or T cells. Now, I put this chart on here. This is normally, if this was a face-to-face class, I would be writing this on the board and would be kind of tackling these two lines of defense. So it's all third line of defense, but there are two big parts to our adaptive immunity. Our whole section on the left is called the humoral branch of our adaptive immune system, and then the whole section on the right, which is the cell-mediated branch of our immune system. So we're going to take it in parts. We're going to talk about some of the parts, some of the different kinds of cells and the receptors, and then we're going to take a look at the humoral branch, and then we're going to take a look at the whole cell-mediated branch. And yeah, there is some interaction between the two. So I'm like, you'll see this come up. I have it kind of popping up every once in a while. Again, I like flowcharts and I like diagrams. And so this is kind of, just showing some basic things and how they're working. So talking about some of the basics, because before we talk about this flowchart, we got to know some of the basic parts that's allowing all of these arrows to happen. So cells do have receptors on the outside that mark them as a specific type of cell. It allows them to recognize other types of cells. It allows them to send chemicals to each other. So all cells in the body do have different markers on them. This is no different. I mean, any cell in the body has different markers. And unlike some of our immune system cells have extra markers. Now, one kind of marker that is found on cells in their body are called MHC molecules. It stands for Major Histocompatibility Complex. That's why we shorten it. MHC. We didn't even know these existed until, based on its name, Major Histocompatibility Complex. Histo means tissue. And it wasn't until we realized that when we started doing organ transplants and tissue transplants that there was this rejection. We're like, it's the same organ, the same kind of cells. Why is there this rejection? That's when we figured out that all cells in our body have these very specific markers, and my markers are different than your markers. And I'm like, my body can recognize all my own. So that's what we realized, that these major histability molecules, complex molecules, are found. So on every single cell in your body, except the red blood cells, so all nucleated cells are going to have at least one of these MHC antigens. But there's two kinds. There's class 1 and class 2. Class 1 antigens, class 1 MHC antigens, are the ones that are going to be found on every nucleated cell. So every single cell in your body except red blood cells have a class 1 antigen on them. That's unique to you. It's why if I gave you an organ, you would probably reject it. Most likely it's because my class 1 antigens on all of my cells are different than your class 1 antigens on all of your cells. Now your cells are going to have other markers, but they're going to have your unique major histability complex 1 antigen on them. Now, class 2 antigens are found on antigen-presenting cells. So these are cells in the body that usually are going to ingest a foreign object and present that foreign object to our T cells to do something about it. So examples of some antigen-presenting cells: macrophages, their job is to eat, it's their name. Dendritic cells that have those long extensions, and even some B cells can be antigen-presenting cells. That they're going to present that there's a foreign object present. Now, just showing an example of why we need that. I'm like, if this is one of your cells over here, this big tan blob cell, if this is one of your cells, from the outside, our immune system can't see inside. But there's a virus inside this cell. So there's got to be some way that our immune system can look at this cell, one of your cells, and know that there is something foreign inside. That it means it needs to get targeted for destruction. Now, how it does that is that one of these the virus itself will have some fragment on it. So this big green virus, there's going to be some fragment from that virus that's going to attach itself to that class 1 antigen, because every single one of your cells has an MHC1 antigen on it, and it will attach itself. It's almost like a little red flag. It's waving like, hey, I'm here. And it's presenting, to some type of T cell. because T cells are looking for virally infected cells, but they can't see inside of them like we can because we're looking at a 2D image, but you can't see inside of it. Instead, those MHC-I antigens are going to present part of the virus so that this T cell knows that this is a cell that needs to die because it is virally infected. Now, just another picture showing class 1 antigen. Again, it's attached right to the cell membrane, and my class 1 antigens found on all nucleated cells in the body. Class 2 antigens are found on some types of white blood cells, the ones we call antigen-presenting cells. Now, B cells and T cells have other receptors. They don't just, they do have class 1 antigens on them because they are cells. They're still nucleated cells, but they do have other receptors on the outside. They are looking for foreign invaders. So B cells have B cell receptors and T cells have T cell receptors, and they look a little bit different. But even though they look different, B cells versus T cells, B cell has so many different kinds of B cell receptors, and T cells have so many different types of T cell receptors that we can literally, potentially recognize billions of different structures and shapes for things that would be considered foreign in the body. Now, we'll take a look at in just a little bit what the B cells and T cells look like. But in the bone marrow, where do we get the cells? Well, the B cells do come from the bone marrow. That's where they develop, but they stay there. And the T cells also made in the bone marrow, but because they don't stay there and they go to the thymus to mature, that's why we call them T cells. But they're the ones that are going to be patrolling. Now, what they're patrolling for is they're looking for some foreign invader. They have those receptors on the outside, and they're looking for a foreign invader. So this is just showing a whole bunch of different cells. Again, they each on the outside are going to have a receptor, and they are all looking for a foreign invader. Now, the foreign invader is this little red triangle. Only one of these cells, out of the millions of cells and the millions of different receptors, only one is specific for this particular antigen. And as soon as it attaches, it forms clones of itself. It's, again, there's usually never just one antigen that gets in the body. It's usually thousands, if not millions, of a particular microbe that gets in the body. And so when one cell recognizes that foreign antigen, it wants a nice strong army. And so it's going to create clones of itself. Now, my note of all lymphocytes have the same receptor specificity. Totally false. I don't even have a comment of bold, but it's false. They all have different receptors. That's where we get the specificity part of our adaptive immune system. Now we're going to take a look at what those receptors look like. So on the B cell, so we're taking a look at our humoral branch, the left-hand side of my one flowchart, a few slides back. But the humoral branch, the B cells, their receptors look like Ys. Now, we call them immunoglobulins, and I'm like, it's the exact same shape as an antibody as well, but the receptor looks like a big Y. It's got 4 chains all together. Don't really care about them. I do care about the very top part, the V, the variable region. And I'm like, I like to think of this big Y is one of those, you see them outside of businesses that flap around in the wind with the big arms. I like to think of the arms, and I'm like, this is the arm, then it's the body. The arms and the hands is where you can vary in the specificity. This particular receptor only can recognize little circles, but it can recognize millions of different shapes, so that variable region, the hand, that can recognize and attach to different foreign objects changes a lot. The other part's considered the constant region because it really doesn't change, hence constant. But the variable part, where the hands are and the different antigens that it can recognize and attach to and activate that B cell, that's going to vary a lot. The T cell receptor doesn't look quite as exciting. It is a double line, but it's pretty straight. No big Y, nothing big exciting. It's like 2 straight lines. It's got a little curve in there. Again, it's going to recognize some type of antigen. And I'm like, usually an antigen that's going to be associated, I'm going to go back a few slides, usually an antigen that's going to recognize something that's attached to an MHC. So here you'd have your T cell. Here's your T cell receptor. It's with the TCR. T cell receptor, and it's looking, that T cell receptor, there's your two lines, is recognizing some type of fragment that's associated with an MHC antigen or protein. Because again, it can't recognize the virus and it can attach to the virus because it's inside the cell. And so your T cell receptor is trying to find something that's attached to an MHC I antigen, not the actual organism itself. Now, there are different kinds of T cells. B cells, although they can vary in their receptors, there's like one kind of B cell. T cells gets a little more variety. Now, how they change depends on the thymus gland and the different types of hormones, but there are different types of T cells. Now, the two major types of T cells. One is the cytotoxic T cells. Usually we just write it T with a little c. And then the helper T cells, T with an H next to it. Now, based on their names, we also call them CD4 cells or CD8 cells, based on receptors that are found on them. But there are more than one kind of cell. We'll talk a little bit about the different kinds of cells once we get to activating the different types of cells. But they work different from each other. Again, and I'm like, it tells you already. I'll just talk about it. Cytotoxic T cells, its name, it's toxic to cells. It's there to kill virus-infected or any kind of damaged cells like cancer cells. Helper T cells, well, they help the cytotoxic cells. Maybe they need to become activated. Some B cells can become activated with helper T cells. Now, where and how the B cells mature. Again, a B cell, it's a lymphocyte, and it just has, like, here's your B cell, the outside of it. Here's the big Y B cell receptor that's on the outside. But B cells, again, we have millions of B cell receptors. So there's only one kind of B cell, but there are millions of different B cell receptors that it could have. Again, they are recognizing very specific foreign invaders and attaching to them. This particular B cell receptor, this big Y, the end of it, that variable region, is specific for this particular cone-shaped structure. And once it attaches, it can activate the B cell. Now, again, all of these receptors, well, whether it's a B cell or a T cell, they are looking for antigens. Again, antigens are any foreign substances that elicit some type of immune system response. Now, antigens, so it's any substance that elicits an immune system response. But antigens can have a lot of different parts to them. And so our immune system can recognize not only the whole antigen, but it can recognize also different fragments of the antigen. I liked this picture I found somewhere. And I'm like, that the whole thing is a foreign object. But the immune system can recognize individual shapes, these epitopes, as foreign as well, which can activate the immune system. Same here, the whole thing is a foreign object, but the immune system can recognize individual shapes as well. Now, this link, when you open up your PowerPoint, you should be able to click on it as well. It's a nice little video, kind of just talking about antigens and different epitopes. If you'd like to watch the little video clip, it's short. Now, we're going to look at what happens with B cells. So we're focusing on this side of my flowchart. It's the humoral branch of the immune system, and we're going to be looking at B cells that have B cell receptors that look like Ys, and the hands of the top part of the Y, that's the variable region that's going to recognize very specific invaders. Now, the main events involved in B cell response. One, well, we're going to bind the antigen, and we're going to have clonal selection, meaning we want to make more copies of that particular B cell. You want to mount your army. We're going to have different chemicals that are going to assist us with this. There is usually some type of T helper cell that's going to help activate. Again, when the two branches work together, they generally work more effectively. We're going to have all of that B cell activation, the full-on clonal expansion, and production of antibodies. We're going to talk about how we make those antibodies and how they work to get rid of the foreign invader. So this is your book picture. It's got a lot going on. I found this one, not quite as much going on. So I'm going to start explaining it this side. Then I'll jump over here. So this is showing 3 different B cells. They're all lymphocytes. They've got a big old nucleus in there. But if you notice, the outside, the Ys, the B cell receptors, they're different from each other. There's 3 different colors. Well, this is an antigen. It's a foreign invader that is going to activate our immune system. But out of the 3 B cells, only one of them, the one that has the dark blue receptor, can recognize this particular one. Again, those receptors are very specific for recognizing very specific antigens. So only this one is going to be able to recognize this antigen. Once it does, it becomes activated. It's, hey, this is the thing I was supposed to recognize. I'm up. This is my job. I need to get rid of it. And so it starts to make clones of itself. It's mounting its army. Now, most of those clones are going to change from a B cell and become something called a plasma cell. It's an effector cell, meaning it's going to do something. It's a plasma cell, most. Now, most of them become plasma cells. This book picture has 3 out of the 4. Most of them become plasma cells, and the job of a plasma cell is to churn out antibodies. And we'll talk about how antibodies get rid of the foreign object, but the job of a plasma cell is to make antibodies. Now, if those activated B cells didn't become a plasma cell, the other option is they're going to become a memory cell, that the next time it sees this little red structure, this little red antigen, it knows how to become a plasma cell and churn out antibodies so much faster. Instead of several days, several weeks, because this could take 10 to 14 days for this to happen, it can happen in 10 to 14 hours. And we can churn out antibodies and target that antigen for destruction a whole lot faster. Now, this picture showing the same thing, but it's also just showing a little bit with the T helper cell helping along with this. So here is a B cell, and it's got little Ys, it's got little B cell receptors all over it, and here's the microbe. Now, anytime this B cell attaches to the microbe, it is activated. However, a helper T cell can also further activate it. And I'm like, and generally when a T helper cell further activates it, it usually gets more memory cells out of it, and the memory cells usually last a little longer. So it can do it without the help of a T helper cell. It doesn't need it, but it's generally better if the cells work together. So again, that activated B cell, most of them become plasma cells, and the job of a plasma cell is to churn out antibodies. Now, if you noticed over here, it doesn't look so pretty over here. Over on this one, if you look at the shape of the antibody, and then if you look at the shape of the B cell receptor, they look identical. And they're supposed to. They are still both technically immunoglobulins, little glycoprotein fragments, but they should look identical because these antibodies that were made are made and shaped so that they can recognize that same thing, that same antigen that targeted it, and activated them to begin with. So antibodies look just like the B cell receptors. So those antibodies, again, they look just like the B cell receptor. They look like big Ys. They have that variable region that recognizes that same antigen that started the whole process off. The top part that binds to the antigen, it's called an antigen binding fragment. I just like that they call it FABs for fragments that bind to antigen. That's the arms, the hand part of the antibody. The body portion of it, it's called the crystallizable fragment. Not as exciting. It's kind of the constant region. It can bind. An antibody body can bind to some types of cells. But generally, it's looking for the antigen binding site. Now, again, if a foreign object gets in the body, like a bacteria or any extracellular fragment, we're going to target the humoral immune system. And it's like, okay, so a bacteria got in, we activated a B cell, we made some plasma cells, then we made antibodies, but we haven't gotten rid of the foreign object yet. Well, the antibody's job is to get rid of the foreign object, and it can do it in several different ways. So these are all different ways, these 5 different ways on how the antibody can get rid of the foreign object. There's opsonization, I'm going to show you the pictures and then describe them. Neutralization, agglutination, complement fixation, and the precipitation. So the arms and the hands part of the antibody is going to attach. It's attaching to all the little pink triangles on here, and it will coat the foreign object, whatever got in the body. Well, one of the ways that these antibodies work is a process called opsonization. Is because the antibodies coat that foreign object, it's going to increase the chance of phagocytosis. There's your big old eating cell, your macrophage. Opsin means phage or phagocytosis. Again, some of these bacteria, if this is a foreign object, if the foreign object is a bacteria, which usually if antibodies are produced, it's a bacteria. A lot of these antibodies have a capsule, which that in itself provides a little camouflage for our immune system. Well, you're not really camouflaged anymore if you're completely covered in antibodies, so you're more likely to get eaten. Some of these bacteria are almost a little slippery, that our macrophages try to eat them and they can't. But you're not so slippery anymore if you're completely coated in antibodies. So opsonization is just the fact that the antibodies coat the bacteria, it increases phagocytosis. Another way they can work is that the antibodies are going to bind and block some critical region or some critical receptor, usually on some type of toxin or on some type of virus. And they're going to neutralize them or activate them, that the virus can't reproduce. It can't get into the correct cell, which means it's not going to reproduce. The toxin can't do damage because these antibodies are coating the part of that toxin that now it can't get into some particular cell and cause damage. So those antibodies are going to block binding sites. And they're going to neutralize those toxins and viruses. Another way they can work, agglutination. It's got the word glue in it for a reason. Agglutination is clumping. Because an antibody has 2 arms and 2 hands, it can potentially then attach to 2 microbes. And because you can have a whole bunch of antibodies and they're constantly all attaching to 2 different microbes and then to each other, you can make these big, huge clumps. Now, by clumping foreign objects together, it increases the chance of phagocytosis. They're definitely going to be more visible, but they could also be a clump large enough that they get filtered out right in the spleen, so they can't circulate around in the body. A fourth way that antibodies work is called complement fixation, that these antibodies are going to attach to the foreign object and they are going to trigger complement proteins. They're going to cause the cell to lyse or to rupture. And if it's going to lyse or rupture, it's going to die. And the last one is that antibodies it's kind of like agglutination, but they're going to clump really tiny antigens. So they are insoluble antigens, so really tiny antigens. We're not clumping big, huge things. We're clumping little tiny things. And again, by clumping those tiny little insoluble antigens, more likely to get filtered, more likely to increase phagocytosis. So the activated B cell doesn't kill the foreign object. Instead, by activating a B cell and making antibodies, the antibodies help target that foreign object for destruction. Now, there are 5 different antibody classes. So although they're all specific for a foreign invader, specific for an antigen, there are 5 classes. It has to deal with their shape, how they hang out together. But there's 5 classes. Now, of the 5 classes of antibodies, they're all named for Greek letters. So immunoglobulin gamma, mu, alpha, epsilon, and delta. I don't care that you note it if you know them, and just if you're wondering where the letters came from. But your immunoglobulin or your antibody G, it is the most common one. So if we draw your blood right now and look for what antibodies are even circulating around in your body right now, the most common one is going to be your immunoglobulin G. It's going to be one that can cross the placental. it's going to be found in your blood serum. So if we drew your blood, we'd look in your blood serum and we'd find it. Your immunoglobulin mu, which I think is the coolest looking one because it's always hanging out in pentamers. So you can have 5 little antibodies. The body portion all hangs together, and so it can do awesome agglutination because they hang out together. You have 5 antibodies, 10 arms, so it can clump a lot of stuff together. But it is the first responder, and so it is the first one that is made. And so when you're activating those B cells to become plasma cells, your immunoglobulin or antibody mu is going to be the first one that's going to show up. Your immunoglobulin or antibody A for alpha is the one we find in a lot of different bodily fluids. So saliva, tears, breast milk, and colostrum. This is why babies can get antibodies from mom, is because they're getting a lot of those immunoglobulin A antibodies. We can even find this particular antibody in semen and vaginal secretions, again, looking for specific foreign invaders. And it's also the one that it works a lot, usually with precipitation and neutralization of different types of foreign objects. Your immunoglobulin E, this is the antibody that's usually popping up most and in highest quantity if you're suffering from an allergic response. It's usually triggered by histamine. And so when you are having an allergic response, you generally take an antihistamine because it's your immune system that's causing all of your allergy symptoms. But we also find a lot of immunoglobulin E in parasitic infections. And so if you have some type of parasitic worm, you seem to have an increase in immunoglobulin E. And the last one, the immunoglobulin D, we find this particular antibody on the B cell, in the same place the B cell receptor is. We don't know its exact function. So we don't know everything about the human body. So it may act as another B cell receptor because they look alike, but we're not exactly sure. Now, regardless of where the antibodies are made, a large quantity is going to end up in the blood, circulating the whole body, trying to find the foreign invader that triggered them anyways. And so when we would draw blood and look in the serum, we can find specific antibodies. It's called anti-serum. It's serum that contains specific antibodies. Now, I already said before, immunoglobulin Mu is the first responder. So the first time you're ever exposed to some type of antigen, that's your AG. We're going to go with tetanus today. So the first time you're ever exposed to the tetanus bacteria or the tetanus toxin, it could take 10 to 12 days, if not more, before you even start to get antibodies made in your body. So it could take a while after the first time you're exposed to something. It could take, sometimes a month before you have full immune system response. However, this is primary response, the first time you've been exposed to something like tetanus. However, the second time you've been exposed to it, you have what's known as an anamnestic response. Here we go. Which means amnesia, without the an, means forgetting. When you put an at the beginning, when you put a or an at the beginning, it means without or not. So it's the not forgetting response, which is kind of the long way of saying the memory response. An anamnestic response is a memory response. The fact that could have taken weeks, if not a month, before we had a full immune system response to something. It takes a while to figure out, how do I make those plasma cells? How do I turn out antibodies? But the second time you're exposed to it, you have full immune system response within days. This is why we get vaccinated. The vaccine is giving us that first exposure. So if in the future you're ever exposed to that same thing, you can mount a super fast immune system response so that you never get potentially that disease you were vaccinated against. Now, to help us make antibodies, because you can make antibodies, target them, but a lot of antibodies go away after a while. We have memory cells that stick around, but a lot of times the antibodies can go away. And there's times we want to make antibodies. And so we figured out a way how to do this. They're called monoclonal antibodies. That's when we want to make a lot of antibodies. We can do this in a lab. They're all of the same kind. So maybe we want to make little blue antibodies, like in my one picture. So we're going to make a pure preparation of an antibody. So generally what we do, this was your book's picture, I found this one because I liked it too, both have the mouse in it, is that a mouse is going to get injected with some specific antigen. I don't know, little red triangles, whatever you want to think of them as. And I'm like, it's going to secrete antibodies. We can mix those antibodies, those plasma cells from the mouse, with a cancer cell. It's called a myeloma. Anything that ends in oma is cancer. So we're going to mix a plasma cell from the mouse. And this particular plasma cell knows how to churn out the correct antibody, these little red ones. So this plasma cell is going to get mixed with this cancerous cell, and we're going to have something called a hybridoma. Now, when you think of hybrid, you might be thinking of car, but hybrid means a combination of two things. A hybrid car is a combination of an electric engine as well as a gas engine, while a hybridoma is combining a plasma cell with a cancerous cell, two kinds of cells, and now we have a new cell. It truly is a cancerous plasma cell. Now, a cancerous cell will continually divide. Nothing tells it to stop. That's what cancer is. But because it's a cancerous plasma cell, this is a plasma cell that will continually churn out antibodies, because plasma cells only churn out antibodies for several days, maybe a week before they die. And so now we've got a plasma cell that will constantly and consistently churn out the correct antibody, which we can use for therapy, we can use it to diagnose diseases, we can use it for other things. Now, we talked about B cell activation, everything in the humoral branch. And I'm like, and B cell activation made the plasma cells to make the antibodies, and we talked about the different kinds of antibodies, and we talked about how those antibodies can work. Now we're going to flip it over and take a look at the cell-mediated branch, the T cells, the fact that we have these cytotoxic cells and the helper T cells. Kind of what are they recognizing, what are they not? My big thing is T cells in the cell-mediated branch is looking for intracellular pathogens. So things that are hiding out inside of our cells, which means they're harder to see. But T cells have figured out a way around that. Is because they are looking for things attached to MHC I or MHC II antigens. So T cell dependent antigens have to be processed by phagocytes called antigen-presenting cells before they come in contact with the T cell, which means we need an antigen-presenting cell to present the antigen to our T cells, because those antigen-presenting cells are going to have those class 1 and or class 2 antigens on them. Here's my little picture. I like it. Yep, this is what it looks like. Here's a dendritic cell. A dendritic cell is an example of an antigen-presenting cell. And it ate this microbe, so it's thinking about it. Like, yum, that's what it is. So it ate this microbe. Now, because it's an antigen-presenting cell, it does have both class 2 and class 1 antigen. All cells in your body have class 1. Antigen-presenting cells also have class 2. Now, the class 1 antigen is going to activate cytotoxic cells, killing cells. The class 2 antigen can present this thing and activate T helper cells. So I'm like, it's just that easy, right? So there are 3 types of cells that can be antigen-presenting cells. So whether it's a macrophage, a dendritic cell like this picture, or even B cells, rarer, B cells can be antigen-presenting cells. Antigen-presenting cells do have class 2. Otherwise, all cells in your body have class 1 and can present foreign objects. Now, this is just showing how we can activate the different cells. So, we have T cells in the thymus. However, this particular cell, it's a CD8 cell, it's going to be a cytotoxic cell, but it's not activated yet. It only becomes activated when, here's our dendritic cell, it's an antigen-presenting cell, and I'm like, it has class 1 because all nucleated cells have class 1. It's presenting this fragment, this little red thing, to this going to be a cytotoxic cell. It's presenting to it, it's going to attach to it, it's going to activate it, and you're going to have that clonal expansion. Lots of copies of itself, lots of cytotoxic cells, And if they don't become additional cytotoxic cells, it becomes a memory cell. However, same antigen presenting cell, we just copied it and threw it over on the other side. Because it's an antigen presenting cell, it does also have the class II antigen on it. And so your CD4 cells, which will become a helper T cell, can recognize the antigen, that fragment. It's going to become activated. Clonal expansion is going to make more of it. And yes, it's going to make some memory cells as well. And I'm like, so it's constantly, we're going to be activating cytotoxic cells using any fragment on a class I. And we're going to be activating helper T cells by anything on one of those antigen presenting cells. So we've got different types of cells to do different types of things. So the cell-mediated immunity is going to require direct involvement of T cells in the immune response. So it involves T cells. But again, before that T cell gets activated, the antigen has to be presented on an MHC complex. So before this T cell becomes activated, it has to be presented, the foreign object has to be presented on an MHC1, they call it a complex, protein, whatever, to be able to recognize it. Because this T cell cannot become activated and recognize a virus that's inside of a cell. It can only recognize some structure that's attached to an MHC1 or 2. Now, once activated, again, it's going to make copies of itself, and it can become effector cells to go kill or go help, depending on what kind of T cells, and it can make memory cells. And so I'm like, now, there are several different types of T cells. The T helper cells, also called your CD4 cells, they're activated by the MHC2. I'm going to go back to this one. Your T helper cells activated by the MHC2. And I'm like, and their main job, again, they're called helper T cells for a reason. And it's because they help regulate and activate to different antigens, but they can also help regulate T cells and B cells. So again, a B cell can become activated as soon as it sees a bacteria, and it can make plasma cells and it can make antibodies. But if a T helper cell also helps activate those B cells, you're generally going to get more plasma cells and you're going to get more memory cells. So it's better if a T helper cell activates a B cell. But T helper cells also kind of keep some of your cytotoxic cells in check. Cytotoxic cells and even some of our natural killer cells down here, they're really good at what they do. They like to kill, sometimes too well, that sometimes they start to kill normal body cells. So T helper cells can kind of keep them in check a little bit. Now your cytotoxic cells, they're activated by the MHC1 antigen. So your cytotoxic cells activated by any cell in the body that has a nucleus, means everything but red blood cells, does have a class I. And so any cell in the body, if there's something foreign in it, it'll get presented on a class I, and it can activate a cytotoxic cell to kill the infected cell. Now, another kind of T cell, it doesn't get anything fancy like CD8 or CD4. They're called natural killer cells. They lack some of the specificity, which means they're really good at killing. They circulate around in the body, and they are usually ones that are first to recognize and kill some of your cancer cells, and they're usually one of the first to also recognize and kill virus-infected cells. Unfortunately, because they can kill really, really well, they do need to get regulated, and so those helper T cells and even some regulatory T cells can help with that. But they're usually the first ones that recognize and start killing until we start to get some of the other cells activated. This is showing some of your cytotoxic cells killing, because cytotoxic cells can also kill cancerous cells as well. A cancer cell is an abnormal cell. Our body doesn't like abnormal anything. So here's a cytotoxic cell killing a dead cancer cell. Now, Mike, and it usually does that. Again, it's called cytotoxic. It produces various types of toxins and chemicals to kill a cancer cell. And so it's going to kill. It's almost going to cause apoptosis or cell suicide. Like, hey, you're infected or there's something wrong with you. It will kind of trigger that cell to kill itself. Now, my little concept check halfway through. The lymphocyte, the white blood cell responsible for recognizing and destroying cancerous cells, is the cytotoxic cell. The natural killer is usually the first one that shows up for cancer, but the cytotoxic cell can start to recognize very unique cancer cells. It's one of our newest immunotherapy methods. You might now start seeing commercials, now that I talk about it, start seeing commercials. And we're using your own body's immune system to fight cancer. We are. We know that cytotoxic cells can target cancer cells. So can we try to trigger our immune system to make more cytotoxic cells for those that have cancer? And our own body's immune system can kill the cancer off. It's having awesome results. So it's, not working for every cancer, every type, but it's working. It's awesome. We're using our own immune system. We're just kind of boosting it up so that it's more effective against cancer. Now, immunity. So once we've recognized foreign invaders in the body and gotten rid of them, we have memory cells. Whether they were memory B cells or memory T cells, we have memory cells. And so we develop what's known as immunity. Now, active immunity means your body has been presented with an antigen. It stimulates making of antibodies as well as memory cells. Takes, weeks if months. But once we have those memory cells, we have a long-lasting immunity. It was active. Your immune system was active at fighting the whole immune system response. Passive immunity means you're given antibodies. You didn't make them. And I'm like, now an example why you might be given a passive immunity. So you could develop immunity. Let's say you got bitten by a venomous snake. Guess what? You have B cell receptors that can recognize snake bite venom, of like any kind of snake bite. It can recognize that venom. It can activate those B cells to become plasma cells to churn out antibodies, and it can neutralize that venom. Problem is, it could take weeks. And if you've just been bitten by a venomous snake, you don't have weeks. And so there are times that you are given antibodies so that those immediate antibodies can bind up the toxic venom. So you're given antivenom. You're given antibodies against that particular antigen. That happens sometimes the last time I heard, is tetanus. I talked a little bit about, tetanus shots or vaccines. But if you haven't gotten vaccinated or it's been a long time, tetanus is a really deadly bacteria, and it's not the bacteria that kills you, it's the toxin that it makes. That again, it could take weeks, if not months, to develop a full immune system response. And if it's the first time you've ever been exposed to it, you might not have that long to live. And so they can administer antibodies. Problem is, we don't generally have stockpiles of it, at least not against tetanus, because it's pretty rare. Now, a natural immunity means you acquired this antigen as part of, you came across it normal everyday life. Artificial immunity is you were exposed to a particular antigen via a vaccine. Now, this is when I would normally poll the class. So, imagine if you were going to raise your hand, is that some of you listening to this right now have a natural immunity to something, and some of you listening to this right now have an artificial immunity to something. So the question is, what kind of immunity do you have, natural or artificial, to chickenpox? Because some in this class have a natural immunity to chickenpox, and some in this class have an artificial immunity to chickenpox. Well, it depends. If you have a natural immunity to chickenpox, it's because you had chickenpox. If you have an artificial immunity to chickenpox, it's because you got the chickenpox vaccine. So it depends. Back in my day, there was no chickenpox vaccine. We all just had to get the chickenpox. So my sister got it first, and my mom made my my sister give me a kiss in middle school, so I would get it. So, we're both sick at the same time. Thanks, Mom. So I have a natural immunity to chickenpox because I've had it. Now, we can put those words together, natural and passive, or the natural and artificial and active and passive, is just some examples of where we get all of our immunity. A natural active immunity is something you acquired in your daily life and you developed your own immune system to it. So natural because you acquired it along your everyday life, and active because you made antibodies. Natural passive means, I was going to say, someone acquired everyday life exposure to some type of antigen, but then passive because antibodies were passed on. The biggest example for natural passive is breastfeeding, is that mom, everyday life, was exposed to something. She hadn't, made antibodies. And I'm like, natural, but those antibodies are passed on to baby in that breast milk. Now, my note about passive immunity is that passive immunity, if I go back, it doesn't create any memory cells whatsoever. It's short term. So only when that infant is nursing actively that they have those antibodies in their body. And there's debates on, different types of antibodies, how long they stay in infants after nursing, but it's not forever. And I'm like, they don't have any memory cells. They've never been exposed to the particular antigen themselves, but they do have some immunity. Artificial active. So artificial, this is all via vaccine. And I'm like, is that we're going to expose the body to some type of antigen via vaccine, and your body is going to recognize it, mount its own immune response. Active. Artificial passive is that via vaccine is that you're given antibodies. So if you were bitten by the venomous snake or you developed tetanus, and I'm like, via a vaccine, you were given antibodies that were made somewhere else. Now, some principles of making vaccines. So there are different kinds of vaccines. Most vaccines are made from killed cells. So we took a living organism and we killed it, or we inactivated a virus, because a virus isn't technically alive, but we inactivated it so it can't cause disease. Some vaccines are made from live cells or viruses, but they're called attenuated, which means we've modified them so they can't cause disease. So a lot of times we shoot them with some radiation, but they're still alive, but they can't cause disease anymore. Some of them are made using different kinds of molecules that came, so different antigens, some of the epitopes that come from the bacteria or the virus, but not the whole bacteria or the virus. And some of them are made from genetically engineered microbes or microbial agents. Now, our checklist on things we would like for our effective vaccine. We would like it to have a low dose with very few side effects and not cause any harm. We want to be protected against, all forms of the disease. You want it to stimulate both B cell and T cell response, so you get memory cells everywhere. We want it to work a long time. We want it to work with minimal doses or boosters. It's annoying to have to go in for your third booster shot all the time with every single thing. We want it to be cheap. We want it to have a long shelf life, and we want it to be easy to administer. All things we'd like of our different vaccines. Now, an attenuated or live but modified virus versus a killed, dead virus. There are some advantages of giving someone an attenuated live organism. The organism can multiply and produce some kind of infection. An infection just means that the organism got in and took hold. But because we modified it, it doesn't cause the disease. But because you have a large number of the organism in the body, because it was multiplying, it's going to elicit a stronger immune response, which means you're going to have a longer lasting protection. So it usually requires fewer doses, fewer booster shots. So it's like, yay! However, disadvantages, it's a living organism. It usually requires special storage. It usually has to get refrigerated. It can be retransmitted to other people. It is a living organism in the body. If you have this organism, it can be transmitted to others. Now, it's listed as a disadvantage, But in theory, if you were vaccinated with one of these, you could spread your vaccine and be vaccinating other people too. Because it is an inactivated organism. It's not going to cause the disease. However, things can mutate. We've already talked about mutations. Things can mutate, and there is always a possibility it could mutate back to that disease-causing strain. That has happened. It's rare, but it has happened. And so a lot of different things we no longer give attenuated. One of the ones we used to give, it was decades ago, we used to give the attenuated or live polio vaccine, but I think it was over in Europe somewhere that it changed. It had mutated back to a virulent strain, and a whole bunch of people that got vaccinated actually developed polio. And so I'm like, since then we just don't mess with it here in the United States. Always the dead or killed version. And so I'm like, it's rare that it happens, but it could happen. Now, people that have a weaker immune system, it could also be an issue, is that you are given a living organism in your body, which means your immune system has to recognize it and fight it off. But if you have a weak immune system, you might not be able to fight off even that weakened version of the organism. So it is an issue. The attenuated is an issue for immunocompromised individuals. They're always going to give the killed vaccine. So this is just showing the difference between that live attenuated or the killed cell. They're whole cells, because it's the whole cell. And I'm like, either one is just killed or inactivated, usually some type of heat or chemical. It's given. Because it's dead, it can't multiply, can't cause disease, but it still has all the little antigens on the outside to trigger our immune system. A live attenuated cell or virus, again, it's made so that it can't cause disease, usually with radiation, and once it's administered, they can multiply, so they stimulate that stronger immune system. Now, we can also do vaccines from micro parts. You don't need to be given the whole cell. Instead of the whole organism, sometimes we just take some of the tiny little antigens on the outside. Maybe we just take part of a capsule that's unique to a particular organism, or some type of surface protein, or some type of toxin. I'm like, and you're given just that small, part of those things. It's enough. It still has the antigen, those different little epitope structures that are going to elicit an immune system response, but you don't have to be given the whole organism. This is kind of showing too, and I'm like, a cellular vaccine. I'm like, this is with the virus, and I'm like, versus the bacteria. You can be given just one small part of the organism without being given the whole organism. You just need the part that's going to trigger an immune system reaction against that specific organism. Now, we can also start to make our own vaccines do whatever we want it to. We can use other cells to make vaccines. This is the hepatitis B virus. Well, we know there's a little piece of DNA in there that codes for a particular surface antigen that this virus has. Well, we can snip out that little bit of DNA, put it in a plasmid, and can put it in another type of cell, like a yeast cell, which is really easy to grow in a lab. And that yeast will actually, because it has that viral DNA in it, start making that particular surface antigen. And we can harvest that surface antigen, put it in a vaccine. It's going to trigger an immune system response so that if you were ever exposed to this particular virus in the future, you would know how to recognize that virus-specific antigen. And then we also have genetically engineered vaccines. And I'm like, that we can make, and I'm like, our own. And I'm like, it's not just putting them in yeast to make different fragments, is that we can take DNA of a certain pathogen, we can put that DNA in a plasmid and just put that right in us. I'm like, so you're given DNA from some type of organism. It means your cells can make that particular microorganism's antigen. So your cells can start churning out little green triangles, which is something that this particular organism would have had. And so your immune system would recognize that particular antigen. It would be able to mount an immune response. You would have memory cells. So if you ever encountered those little green triangles again, you'd be able to fight it off. Now, It is our newest area of research. So right now we have experimental vaccines. I'm hoping soon they're not in the experimental stage, but we have experimental vaccines for Lyme's disease. That would be amazing living in the area of the United States we live in these days. Experimental vaccines for hepatitis, herpes, the flu. We might actually have a flu vaccine that works against all strains, even when they change. Tuberculosis and malaria. Again, it's if we can find that little snippet of DNA of one of these organisms that we know is unique, and our cells can make that unique structure, and then if we're ever exposed to that unique structure in the future, that specific DNA, that we will to fight it off. Awesome stuff coming up. Now, last little concept check. And I'm like, if only a portion of a pathogen is being used as a basis for a vaccine, and I'm like, what would it be called? It would be the acellular. Is you're just, it's not the whole cell, because A means without. So without being a cell, it's just a small little fragment. Now, administering vaccines and some side effects that can come from vaccines. Most are administered by shots. A few of them are oral or nasal. I know the flu is now nasal. Some vaccines do require something called an adjuvant. It's a compound that's added to increase the immunogenicity, meaning how effective it is at activating our immune system, and how long that vaccine and that triggering of our immune system lasts. Now, my best way to compare, what an adjuvant is. An adjuvant is my morning coffee. It's what wakes me up. It's what keeps me active. And so when we add this adjuvant to a vaccine, it wakes up our immune system. Like, hey, we're going to be exposed to something today. Get yourself ready. And I'm like, and it keeps the immune system active longer. So you're going to have a stronger immune system response. Now, there's a lot of stringent requirements for developing vaccines. I know we are pushing through to develop a COVID vaccine, but there are some very stringent requirements for us to develop any kind of vaccine. We want to make sure there is more benefit than risk factors, because there are side effects for some vaccines. A lot of times there's a reaction at the injection site. Some people will get a fever. Again, your immune system is at work, and so having a fever is not uncommon with any kind of vaccine. Some people might have an allergic reaction. It's rare, but it can happen. Really rare that something can mutate back to a virulent strain, but that's only with the attenuated vaccines. Or there might be some type of neurological response. Rare, but there can be possible side effects. So when known, and I'm like, if we know there's some type of side effect, all vaccines are altered or withdrawn, so they're not given until we can figure out a way to make it so those side effects are lessened or gone, if possible. Now, another thing that is also talked about with COVID-19 is herd immunity. Now, herd immunity really means that if enough immune individuals are out there, meaning they've already had it, they've developed immune system response to it, if enough immune individuals are out there, means they're not going to be able to harbor the virus, we'll talk virus, and it means it can't spread. So it's less likely than a non-immunized person would even encounter it. This is just showing, when a vaccine, like COVID, is exposed. And I'm like, right away, if no one has immunity, everyone's going to get it. I mean, that's just kind of how it's working. And I'm like, or at least a large number, not everyone, is going to get it. But if some people, and I'm like, and it's going to spread, that's my little area, the little connectors, and it's going to spread. However, if some people have immunity, they are now my black lines, they can't spread it anymore. So if they're exposed to someone that has it, it encounters a dead end. They can't spread it anymore. And so less people are going to get it, because if this person can't get it anymore, it's a dead end. These people are protected that are hanging out with these people. And if you get enough people that have had it already and are now immune, it can't spread anywhere. These 3 people that just got it, who are they going to spread it to? Because all these have had it already. And so these other people that haven't become more protected. So I know they're talking about, what's going to help with COVID-19 is if we develop herd immunity. My warning, though, with that is that for herd immunity to take place, they figure at least somewhere in the 55 to 82, and that totally ranges depending on how easily a virus spreads from person to person, the contact transmission. And I'm like, 55 to 82% of the entire population has to have had COVID-19 or gotten vaccinated against it. But we don't have a vaccine. So for herd immunity to get it, that means if we go on the low end, we'll say 50%. It means 1 in every 2 people. That's millions of people all have to get COVID-19. So everyone's like, we need herd immunity. But if half the United States gets COVID-19, unfortunately our hospitals and medical facilities can't handle that much. I know not everyone gets hospitalized, but if we're talking half the United States, and that's on the low end number, like lower than the low end number, and I'm like, our medical system can't handle it. So maybe we'll get there, hopefully herd immunity can happen, but it's not going to happen fast. And we don't want it to happen fast. Hopefully we'll get a vaccine. So we're going to end right there. And I'm like, let me know again if you have any questions though on this chapter. I know there's a lot of information in here. Not a bad idea to listen to the PowerPoint twice, kind of skim over the PowerPoint because there's a lot of key topics in this chapter.